2 microphones in the audience, and we'll go to a question and answer session where we're about 10 minutes behind. Then we'll try to go for about 30 or 40 minutes. And if, thank you. I think we've got microphones here on the tables.
You're over there.
And we could take, so we have Walter Katownik with 1 microphone, and Doris Walsh, I think, with the other. So if you have a question, please raise your hand.
Question regarding the IFSHD study. If they weren't officially diagnosed by age 10, but were showing symptoms before then and have been genetically diagnosed, my son is 19 now, are you looking for younger kids, or is he available to participate in that study?
Yes, I think the age of the patient at the time enrolled in this project can be any age, as long as he has a genetic diagnosis and also shows symptoms before 10. Yeah, but if you're interested, you can always go to one of the sites, and the clinician will actually explain more. Mm-hmm.
Dr. Jones, question for you. So can symptoms be turned off because of environmental conditions, from your perspective? So you said that there are different things like diet, sleep, stress,
Mm-hmm.
what you eat, prenatal care, all those things can be indicators or can impact
Yes.
how someone develops the disease or doesn't develop the disease.
Those are environmental factors that can impact the epigenetic status, just as in epigenetics in general of the genome. Whether any of those are particularly involved with FSH is not known.
Yeah, so I know you don't know the answer to this, but I'm asking your best thinking, if you had to guess. If you had a son who's 12 years old who had developed this condition, and you thought there was a chance that some symptoms could be turned off at some point in time that had originally been turned on, what would you do?
Oh boy. So from the epigenetic standpoint, most of the initial establishment of epigenetics is going to be developmentally, and so this initial state's already been established. And so now the trick is there anything you can do to kind of to switch it back as an adult? And I think that the best approach is really just going to be small molecule approaches. I'm not, the lifestyle change, I think sleep and stress and those types of things, are good just in general to take care of yourself. But in regards to really flipping it, so the key about an epigenetic switch is that once they're established, they're very stable. And so I think that these are factors that differentially establish the state. And so flipping a stable, established state is going to actually be pretty tough naturally. I mean, the epigenome is set up specifically not to do that. And so, in fact, so the small molecules, for epigenetic small molecules, though, that can target these epigenetic states that are being developed, one of the nice things about that is if you can flip the epigenetic state, the cell should remember that it's flipped, and it should be a permanent state reversal if you can do it. But right now, I think for epigenetically, the best approach or the best hope is going to be the small molecules that are being developed to target the epigenome. It's just how it is.
Dr. Jones, your description of FSHD as an epigenetic disease seems to describe what others were calling a variation in penetrance of the disease. Is that correct?
I think it explains the variable penetrance, yes.
And so my second question is it an established fact that FSHD is an epigenetic type disease, or is that a theory?
I believe it's established. Silvère, would you like to comment?
At least it has a very strong epigenetic component to it. I think it's a genetic disease because you need a contraction or a mutation in SMCHD1, but immediately the downstream consequences immediate from that are epigenetic in nature.
Right, the genetics are permissive, right? I mean, you need to have,
Yes.
definitely have some genetic requirements. It's not purely but to me the epigenetic state dominates you need the genetic requirements, but the epigenetic state is what determines after you have the genetic requirements.
Yeah, I would agree.
Dr. Jones.
Oh, sure, okay.
Okay. Can you go more into the molecular therapeutic targets that you had shown on your slide, please?
Sure, so the epigenetic changes are, one we assay is, so are you talking about the epigenetic targets or just general targets on the whole, all the targets on the targeting slide? Sorry, just epigenetic targets? So DNA methylation and the overall is really a readout of the overall chromatin state. As Silvère showed his slinky is actually wrapped around full of proteins that are differentially modified. And there's enzymes that make these modifications towards more repressive and more active. And one of the things has always been a problem with epigenetic drugs and epigenetic targeting has been your entire genome, all of your 6 feet of DNA are wrapped and have these marks. It just happens, though, that D4Z4 is kind of distinct. There's a special DNA methyltransferase that targets this. There's actually SMCHD1 that targets this. So it actually stands out from most of the genome as epigenetically distinct. And I think that small molecules that target histone methyltransferases, DNA methyltransferases, chromatin remodeling proteins, as we get into the things that establish the chromatin at D4Z4, will actually be somewhat specific. They're very, actually quite, I believe, specific to D4Z4, and you'll be able to avoid some of the genome-wide effects of altering the epigenome. And so it's just one of those, again, the FSHD is somewhat confounding. It's very complex, but it turns out the complexity is going to be used to the advantage because a lot of it stands out from the rest of the genome. This region really wants to be off, in my opinion, in the primate genome. Really wants this region off, and they developed a way to evolved a way to do that. And I think that there's going to be a number of druggable targets there.
Perhaps I can, right here, Peter. Perhaps I can ask a follow-up. So you sequenced 14 families looking for modifiers? Is that, I think it was...
No, that's comparing asymptomatic to symptomatic.
And in
so
that, are you finding any evidence of a loss of function in a demethylase, perhaps?
We don't have any of the data available on that.
Okay, so we do have some questions from the interwebs. The first had to do with CRISPR, which is a way of modifying the genome. They just wanted to know if there's any activity within the FSHD research world to use that. I'll start with that one.
Maybe Dan's the one to address this, actually. The question is the FSHD research field utilizing CRISPR technology? Is the FSH Society funding CRISPR work?
Actually, there are several projects ongoing. Dr. Kaima, you're in the audience. Michael Kaima, raise your hand, stand up. Dr. Kaima has an NIH grant on CRISPR-Cas9 on FSHD. He'll be speaking later. I believe Dr. Wagner or Wellstone are using CRISPR-Cas9. Louis Kunkel is talking to folks at the Broad Institute and also at Harvard Medical School, people who actually invented Talon, CRISPR-Cas9. So there's a lot of activity going on. The primary people that are funded right now that I know of are Dr. Kaima to NIH, And the FSH Society has 2 grants. We issued a special RFA on genetic engineering. One grant went to Dr. Kava, and another one went to Julie Dumanceau in Paris, France.
The second question just has to do with the DNA testing, and they wanted to know whether or not current DNA testing can clearly and conclusively differentiate between FSHD1 and FSHD2.
I guess that would be me. Yeah, the current testing that is available also here in the United States allows for the differentiation between FSHD1 and FSHD2. Yeah.
Great. And finally, the third is an exercise question, so I'm not quite sure if you guys can tackle this, but it's just another of the ongoing questions of, does exercise help? And this one in particular is sort of extreme exercise.
Perhaps we can involve Dr. Van Engelen, who is here in the audience, and perhaps he can answer this question.
Can you bring the microphone to Dr. Van Engelen? Thank you. Sorry about that. Took me a while to figure out what that sign language was.
Tomorrow we will speak about the trial we did in Holland, training FSH people, and strength training was not beneficial, but aerobic exercise was, and the data and details will come tomorrow.
Thank you.
For some of your studies, you were looking for blood samples from affected individuals and their families to assess the penetrance. At one point when we were involved, it was not particularly user-friendly. And I'm thinking if this affects 1 in 8,000 people, and certainly the families of people who are here, you could probably get lots of volunteers from family members if it were easier to get the blood samples to the research centers, which are scattered around the country. If there were a way for people to just go to their local lab and get whatever blood drawn in whatever kind of tube and spun or processed or whatever and sent to you, you could probably get more samples than you'd know what to do with. So I'm wondering if there's a way to make it easier, more practical and feasible to get the samples that you need from more members of affected families so that you can do more studies.
So maybe Catherine Wagner would answer that question, because...
Well, thank you. That was a perfect opening for us. We actually are going to try to use this venue for exactly that purpose. So both this afternoon and tomorrow afternoon at 2 p.m., there'll be a room if you'd like to come and learn a little bit about our study and sign a consent form. Just do a very simple examination and give us a sample of blood. We're interested in you, and particularly if you are a member of a family in which there is some divergence in the severity of FSH. You've heard from several of the investigators about how there's a real effort to try to understand why some family members are more severely affected than others. And if we can understand that better, and Silvère has identified one modifier of FSH, and if we can identify others perhaps, and if we can try to target those, that could be a path forward for therapeutics. So today at 2 p.m. and tomorrow at 2 p.m.
I think this goes out to all the scientists. Could you please clarify the number of affected individuals? Because a lot of the research, the papers that are out say like 1 in 8,000 are affected, and then 1 in 20,000 are affected. So.
Well, I think Dan mentioned that paper came out today that 1 in 8,333. Is that correct?
Yeah, Dr. Van der Maarel and Dr. Van Engelen had a paper published yesterday. Congratulations, long overdue. We really appreciate it. And it basically changes the number from 1 in 20,000. So Holland, you have to understand, Holland has a really outstanding patient registry. It has an outstanding history with Dr. Padberg and Dr. Rune France. And Dr. France, Rune France is Silvère's mentor, and Dr. Padberg was the early neurologist in Holland. And Rune France and Padberg started working on FSHD, and they had the opportunity to drive and see all the patients in such a small country. Now, Holland has an exceptional registry, a national registry of patients and also genetic materials. So they were the first to come up with the gold standard in 1980 of 1 in 20,000, based on a really good foundation. Now, you can't necessarily extrapolate that to the entire world, but they revisited their data, and I'll turn it over. But yesterday, they came out with 1 in 8,000, 1 in 12 in 100,000, 1 in 8,333, which makes it the most prevalent dystrophy as far as I'm concerned. I'd like to let Dr. Van Engelen, who's the lead author, there, comment more on the paper, but that's the relevant background.
Yes, we use 3 different cohorts of FSH data in the Netherlands, and by combining them in the catch-recatch method, which is a statistical method, we were able to find this number of 1 in 8,000 people. So, because this method also addresses the people that you miss, it is a statistical method. It's also used in ALS epidemiology, and so it is a quite reliable method and to detect the people you are missing in a one cohort study.
Hello, I'm Mrs. Janet from France, ASM Association. So you'll excuse my poor English. I wanted to ask Dr. Jones if epigenetics could explain why FSH is non-symmetrical and why some muscles are affected and some not.
I believe it can explain the asymmetry, in my opinion. I'm not so sure it explains the why certain muscles are affected. I think that'll be a combination of genetics, genes, gene activity. It can be cell type specific, so it'll be a combination of genetic and epigenetics for the affected muscles. But I think the asymmetry can be explained by epigenetics and the establishment of the epigenetic states early in development. In my opinion, that's my hypothesis.